Synthesis and Characterization Lab, Institute of Advanced Technology, University Putra Malaysia. So before we hear the interesting talk by our guest speaker, let me invite the director of ITMA, Professor Dr. Muhammad Nizar Hamidon, to give brief welcoming remarks.
Thank you, Dr. Chacha. Assalamualaikum warahmatullahi wabarakatuh, and very good day for all of you. It's a very my pleasure to welcome all of you to the first virtual ITMA webinar international guest lecture series. This is the part of series of online webinar sharing session for international subject matter expert on nanotechnology. It's aimed to providing key input and share insight of research related nano conduct overseas. So basically with that, I hope that we can use it as one of our method to shaping the overall quality and standard in tertiary education in Malaysia related with nanotechnology. ITMA will be conducting a series of visual webinar international guest lecture meeting after this, alright? Which means it's a continue program, as it is critical to ensure the continuation of our research and to boost university ranking globally to be one of the best institute and universities. We believe that the international guest lecture can add international exposure and in-depth experts toward the research process and experience. Today, basically, we are very lucky to have Professor Dr. Syamir Iqbal from University of Texas, Rio Grande Valley with us. Thank you, Prof, for take some of your time to give this talk. I know that you just have your iftar. Okay.
Yeah.
So
okay.
Thank for that. For it's very our pleasure to have you here. So basically, he will share with us about the nanotechnology and cancer detection. As we know that fight against cancer, half of the battle is won basically on the early detection. All right. So basically, there is some issue for the nano diagnostic. So with that, with the expertise from Prof. Iqbal, which will share with us how that by using the nanotechnology will increase the sensitivity for the early detection of disease. All right. And then, by the way, to treatment of the cancer treatment.
I hope the participant please mute your please mute.
Okay,
Prof.
Yeah, you also mute me. So we all understand the importance of science and technology and innovation in our day-to-day life and the way in which we are transform the world. We believe that the person will have a new idea, alright, related with the nanotechnology, alright, and then we confident that you all will find new ideas and fresh energy towards this webinar. Finally, we wish to thank you all for this today, and also for our international guest speaker, Professor Dr. Syamir Iqbal, everyone for your participation and your commitment, which will demonstrate by your joining us from different part of the world in different time zone. So with that, I hope you have a pleasant webinar today. Thank you.
Okay, thank you so much. Thank you so much, Prof. Nizar, for the welcoming remarks. So now I would like to introduce our speaker. So our speaker today is Professor Syamir Iqbal from which is professional engineer, received early education's Bachelor of Science Electrical Engineering, NED University of Engineering and Technology, Karachi, Pakistan in 1996, and then PhD in Electrical and Computing Engineering from Purdue University, West Lafayette in Indiana, USA. And if when I go through Prof. Samir Iqbal's CV, so I think that he is one of the great researchers. And then I found that currently Prof. Iqbal is the founder for STEMatters, Arlington, Texas, USA, as well as the professor in electrical engineering and School of Medicine, University of Texas, Rio Grande Valley, and having a vast experience as a leader and received many awards, such as I just like pick the one that I think quite interesting, the Career Award National Science Foundation for Nanoelectronic Microfluidic Biochip for ultra-sensitive detection of selective protein biomarkers, and receive a few honors, such as Fellowship for National Science Foundation Summer Institute, Cancer Nanotechnology Analysis, Imaging and Treatment over Multiple Scales at the Methodist Hospital Research Institute, Houston, Texas, USA in 2011. And Prof. Iqbal also having a lot of funded projects related to cancer, sustainable energy and water, as well as interdisciplinary training of nanotechnology for cardiovascular and lung diseases in North Texas. And so far, I think Prof. Iqbal also involved with many community services and causes, and I think maybe later Prof. Iqbal can also share with us in terms of the STEM matters. So maybe some of the lecturers can apply to be the mentor, right, Prof? So maybe for the STEM matters, maybe you can also share with us later. So with that, I would like to invite Prof. Iqbal, Prof. Samir Iqbal, to share the presentation today entitled Nanotechnology for Early Cancer Diagnostic. So with that, the floor is yours, Prof.
Thank you. I really appreciate this opportunity, and thank you, Dr. Che Azurhanem Binte Che Abdullah. And thank you, Director Professor Nizar. I really appreciate it. And I see, mashallah, is a big number here. I will be sharing some of the work that we have done in past, some of the things that we are planning to do, or we hope we could do. You know, the whole world has changed, so priorities are changed. But nevertheless, there are still many interesting things that can be done at the intersection of nanotechnology and fundamental science, whether that's physics, chemistry, or and medicine. And I'm not new to Malaysia. I have, in good days, I used to travel maybe twice every year to KL and every part. And I see I see professors here in this room that I met in my, Professor Khairuddin is here, who I met in Perlis a few years ago. So this is, I know, very good community here. So let's get started. So as you were saying, that I have background in engineering. And when I started my PhD, I decided to get more into interdisciplinary work. And the simple reason is that there is a lot more impact that you can have, because there are bigger problems that are still needing to be solved, like cancer detection. But before we get into details, let's look at some of the things that have enabled us. So I give the example that nanotechnology and nano devices or nanoscience is growing. It's been growing for last 25 years. And it has grown as much that we can have billions of transistors on a chip. And I give this example that there are about 8 billion people on the face of the earth, right? And the numbers continue counting. Some say 7.8, about 8 billion people. And if you look at one small piece, 15 centimeter cube of human brain, that has 10 billion neurons. So think about it, those are cells which are conducting charge, which are conducting data, and they are transferring information. So that's the kind of density that nature has. But now if you look at the devices, the chip industry, it has progressed a lot. Can somebody tell me how many transistors they're making now on a transistor chip? You can type your answer or you can say the number.
Yeah,
How many?
there is a quiz from Prof.
You can type in chat maybe how many transistors or devices they are putting on a single silicon chip that we are using.
Anyone would like to try? Maybe I would like to ask students to try how many chips, how many transistors on the chips.
So it's, if you think about even 2 years ago, they came up with this EPIC ROM device, AMD, which is 32 billion transistors. And the number is growing, number is going up. This is something, at this stage, even the chip industry is at backlog. They are not being able to deliver all the chips that are needed, and they are creating that kind of transistors. 32 billion, that was 2 years ago. And just now, there was a few months ago, 2-3 months ago, they have shown more than 1 trillion transistors on a chip that can do lots of AI work on it. So think about it. And what's the size of the chip? It's about maybe 2.5 centimeter by 2.5 centimeters, which is like an inch by inch. So this is amazing how far we have come in terms of making these devices. Not just making them small, but making them more robust and more dependable. Now, this is where semiconductor industry has been progressing and continue doing more and more into what we call now cloud. So for cloud computing, they're using those engines for image processing, for AI. Where does that lead us? As engineers or scientists or practitioners at interdisciplinary scales, if you look back in last century, first 50 years, where vacuum technology came and changed dramatically our radios, radars, all the equipment that we are using, that dramatically changed. But next 50 years was the transistor age, where things changed, and we, instead of having those big picture tubes, we got thinner devices and smaller devices. And not just that, we got devices that have internet on them, so which are connected with the outside world. So this picture I got from IEEE Spectrum magazine from a few years ago, and they say the next 50 years, that we are in 21 year already in it, they say it's going to be nanorobots and control of organic molecules and wearable wireless internet appliances. A lot of it is true, but I believe much more impact has been coming, and it will come is from biotech, from nanobiotech, where we can look at maybe the genetic makeup of an individual. Maybe we can look at what kind of diseases somebody can acquire from hereditary resources, where we can look at individual cells and play with cells or subcellular entities like proteins or biomarkers, and we'll talk about that a little bit in a bit later. And then an area which is already seeing a lot more work, which is personalized diagnostics and therapy. And we know that for insulin, we have those kind of pumps or pacemakers. But something which is becoming much more recent is the hybrid biomaterials or biodevices, or even printed organs, where we can take a person's stem cells and we put it in a matrix and we can grow them and make them take the shape. So all this is good, but something which is happening is we are still lagging behind on some of the things, which is the sensing of diseases, looking at diseases at genetic, at proteomic, or at cellular level. And when I say cellular, we are talking about few cells. So where we can look at a small population, should be able to see what is the behavior. It seems simple, right? We can take some cells and we can just look them in microscope. But the problem is a little bit more difficult, because we are interested in looking at the bad cells, disease cells. And those are not easy to find, right? So we, like if an individual, we can get scanning done for, we can look for things in our blood, right? We can look for if there is high sugar level, right? We can see many things in blood. We can look at liver related molecules. So that's where we want to do more advanced, and especially more for cancer. And the reason is simple, that we want to make those devices that can take small volume without doing much intervention from users. We want to do things very fast. So here's an example. This right-hand lower side is a PCR machine. PCR machine that used to be maybe 15 years ago were used in labs, but now we are doing PCR chips, which is the other example, which is like a 10 cent in US, a very small coin, which at that size, if you are doing the same process which we are doing with big machine, we can save a lot of cost and a lot of time in getting to the answers. And this becomes important because for us, finding the solution to the early diagnosis has the key for therapy, effective therapy, especially for diseases like cancer. Now, this is an interesting data. If you look at the mortality rate due to different diseases from this is 55 to 2011, you notice that even though mortality rate has dropped significantly over the time, it's still leading cause of death because of cancer. And the cancer overall, it's the second leading cause. And within that, it's the malignant cancer, the cancer that spreads. So yeah, for cardiovascular diseases, we know that there are many affordable screening tools there. We have dietary habits that we know that can help us to have better life for our heart. We have lifestyles. So we can, it's possible to detect them or control them at early stages and cure them. And in some cases, even prevent heart diseases, right? But there are no similar affordable diagnostic platforms for cancer detection, or especially in an early stage. We have maybe one or two tests that are done, but if you look at the whole range, we are still way behind. So hence the death toll from cancer is way high. Now, if the cancer can be detected and localized at the early stages, for a lot of cases, so there are surgical or non-invasive methods where we can remove the cancerous tumors or part of those, right? So now there is the reason why we have not been able to do, because there is no apparent system at early stages. And by the time it's perceivable, or by the time we can find it, it's already too late. And it's especially for this malignant ones, it has spread over the whole body. So this is the major challenge. And if you think about it, we would say, okay, if there are too many mortality, then we may be investing a lot of money in that. So that's another problem. But this data is from few years ago, and things have not changed. Especially maybe COVID has changed, where COVID is getting more funding and more attention. But across the board, cancer has been a bigger killer, but in low to middle income countries. But the funding for cancer research has been much smaller than malaria, TB, and HIV/AIDS. So that's where we need to think about another way of looking at, another cheaper ways of looking at this problem of detection. And this is the motivation of this work, or the talk that we are the things we are going to discuss today. And when you say early detection or minimal invasive, some cancers, they have cells that detach from that primary tumor, and they go into the small body vessels where they can then get into bloodstream. We call them circulating tumor cells. Now these, or in short, we call them CTC, circulating tumor cells. So these circulating blood going in the body that can take these cancer cells along until they find another location, and they get then stuck into that another small blood vessel, and they in the capillaries, and they can start growing. But this process is where we can break it. If we can possibly detect those few cancer cells that are traveling through bloodstream during their journey of going through, that can be a big game changer. But it's not simple. It's a complicated journey for those, and many of those cancer cells, many of them don't survive, but probably there are few that do survive, and that's where we have an opportunity to look for those. And once we have them, then we can do the therapy, which may be chemo, radiotherapy, or whatever the standard things are. Now, this is where we think that there can be a possibly good intervention to look at this. But now, how do we differentiate, or how do we detect a cancer cell from a normal cell, right? So that's where, again, there are some inherent differences. A big difference between them is what we call biomarkers. So, for example, here's just a cartoon of the cell, which is just showing that there are receptors on the cell surface, and we know for many of tumor types, there is overexpression of certain biomarkers. And biomarkers are proteins. Biomarkers are molecules who do some functions in those cells. So here I have highlighted EGFR. Among many biomarkers, EGFR is a very common among many cancers, and this is EGFR is epidermal growth factor receptor. So it's a growth factor receptors, which means if there are many more, so the growth cells will get many more growth signals. Of course, the growth will, we know that cancer cells grow much faster than the other types of cells. So that's an attractive biomarker for the diagnosis, for prognosis, and for monitoring post-treatment conditions. And for us then as engineers, that was an interesting thing. What can we do with this biomarker, which is available on the surface of the cell? And if you look at literature, there lots of work has been done with microfluidic detection techniques. So microfluidics, we know fluids flowing through pipes, right? Water flows in pipes in our house. So it's a fluidic system. But microfluidic is if that pipe is at micro scale. And that's something we all have, right? So our bloodstream is what is a microfluidic system. The further away we go from our heart, the capillaries become thinner and thinner, smaller and smaller. So that hence it is kind of a microfluidic system. So microfluidic systems are already used in research and in industry also, where we look at different properties of cells. Maybe we look for the dielectrophoretic properties. So dielectrophoresis is where it goes to permeability concept, where and a body, if it can readjust the ions in it or the charges, it can become polarized. So that polarization can cause an entity to move differently in electric field. So cells, they have ions all around it, inside it, and especially cancer cells, which are a little bit acidic in their environment. So that can be used to separate them. There are ways where there's magnetic particles have been used to attach with them and then bind them. There's a surface-based binding, right? So where we can coat a surface and use that surface to attach and bind those things. And there's been single cell kind of things where we use capillary tubes and suck out the wall of a cell, and we look at the ionic exchange happening there. So there are many ways, and these have led to the idea what we call cell-based diagnostics, where we can start with a small volume, and on the chip we can separate things out and then detect them in a chamber. And the idea is that all the way from taking the sample, a very small amount, within few hours we should be able to detect the presence of those biomarkers. But there is now the second level. The first level of problem is that cancer cells are too few in bloodstream. Now the other is that they have these biomarkers which can be used to detect them or separate them. And how do we do that? If you think about it, if you have a chip surface and you bring something in contact, they're going to just get washed away unless we make that surface selective or put some molecules on top of it. So this is an area which is called surface modification or chip modification, or there are many ways how we approach it. And there are many ways how it is done. We can do the etching of the surface to change the properties. We can put metal films on it, or we can put molecules and do a self-assembly kind of deposition of those things. And there's a whole range of things, but whatever we do, the idea is that we need to get these entities or molecule-like things that can bind to that biomarker. So if we look at literature, if we look at people who are working on those infections, we can find lots of things. So we do, many people do antibodies and antigens. So they do a sandwich assembly where the surface is coated with an antibody and that selectively binds with an antigen. And then there's a whole kind of detection assembly that goes with that. So in that case, when these entities come, they come in contact, so they stay bound, they don't get washed away. And that's the idea which has been employed. So we have done not just binding, we have done a little bit more, where we have done nanotexturing of the surfaces, which gives us very high selectivity to remove tumor cells from a flowing solution. And that's where we can not just capture those cells, but then go a step further and look at the behavior of the cells. Now, what is that behavior? So that behavior can be, we can look for size differences. And that's the idea of micropores, using very small pores, microscale holes in a membrane, where we push cells through and we look at size difference. We can look at the elasticity difference of the cells, of tumor cells. Now, I may not be able to go through all of these things, different things that we have done, but I'll share a few of those with you. But this picture tells us that what may be different ways how we can do early detection. So the way it's shown, where we, in the size-based resolution, it's like a filter, but it's more than filter, because with one micropore in a membrane, we can measure ionic current across that pore. And then we can push cells through, and once a cell goes through, we can measure the difference in the elastic behavior of that cell. And that can tell us if the cell is disease cells or a normal cell. Now, using the charge is another way in microfluidic channels, where we can separate them based on their charge, or we can detect each individual cell and see what kind of charge it is carrying, again from electrical data or capacitive data. So there are different ways how we do that. We can deform one cell as it is going through, where we can, as it is going through that channel, we can stop it and stop the flow, and then we can measure how fast or how slow does it go in response to that residual pressure that's there. So here's a few examples where we do imaging. So now imaging is something interesting. Now, where we capture those cells on the surface, and then over time we can see the behavior of the cells. So this is a video which we have on YouTube, where we can see the cells changing their boundaries over time. So here, lower picture is showing that one cell taken the images across about every 30 seconds. And what you can see is that cell is continuously changing. So this is something we found a few years ago, that can be used as a detection modality for a cell, for especially metastatic tumor cells. And what it does is, if you look at the top two pictures and lower two pictures, these tumor cells are growing and they are moving and they're trying to make connections, versus the normal cells in the lower two pictures, where they are not moving at all, or they are not making very distinct movements or changes. So this is something which led us to look at it more closely, that what may be the cause of that behavior. And this went back to something what is known as metastasis. So in metastasis, what happens is, as shown in this figure, so the red cells, the tumor cells, they have to go through a level of structures in the body before they get to the bloodstream. And on the other end, when they have to get back to create secondary tumor, they have to again go through a bunch of body structures. One of those structures is called basement membrane, and these top figures are figures of basement membrane. In TEM, if you look at it, we see texture, we see nanoscale texture on the surface of that basement membrane. This is a very interesting thing to see that our body has many different levels of texture, and it has microscale texture, it has nanoscale texture in it. And we looked further and we found out that it's known that nanotexture exists in nature all around us. Here are a few examples. So, for example, lotus leaf. If you put the leaf in an electron microscope, it shows nanoscale structure to it. And they give them, those leaves, super hydrophobic properties. They don't get wet. If you look at watermelon leaf or rice leaf, so they show similar structure and properties which have nanotexture in it, and they show hydrophobic behavior. We look at the wings of a butterfly, right? So it's one material, but we get different colors. So colors come because of this periodic structures in nanoscale, which cause the constructive and destructive interference of light, gives us different colors. So a whole range, peacock wings, peacock, looking at this mosquitoes who travel, who sit on water level and they don't drown. Looking at the spider which is traveling on a surface, right? So gecko, they all have nanotexture. This is something which then led us to think more that what is the relationship of metastasis or metastatic cells that we can use with nanotechnology, the behavior of metastatic cells that can be looked at in terms of their interaction with nanotextured material. And one thing we did for breast cancer, for example, so we worked in many different kinds of cancers. What we see here is a lot of majority of deaths happen because of metastasis in breast cancer, which means that they take the biopsy sample and somehow they miss it because the systems are not sensitive enough to find metastatic cells. So this then leads to the idea that can we have a chip surface where we can create that kind of nanotexture, which we saw for the basement membrane, and increase the sensitivity and selectivity of binding, which means if we create the nanotexture, few things happen as from physical point of view. One is that you will have much more higher surface area, effective surface area, which means you can put down many more molecules for capture of EGFR, for example, what we are looking at. Now, if that happens, which means our surface can be much more receptive for binding. And this is what we did. And in our case, what we did was we used a molecule called aptamer, which is known to be very selective in binding with EGFR. So aptamers are nucleic acids who are known to be, who are experimentally found that they're known to bind to biomolecules, and they do it by changing their shape. So it's a very specialized area, and they use a process called CLEX. So what we did was we took a known aptamer, which was known for binding with EGFR, and that's where we functionalize our chips with nanotexture in it. And this work was done on PDMS, we did it on glass, we did it on silicon chip, a whole bunch of materials, and comparing their binding and behavior, if that can give us much more binding capability. And this is exactly what we saw. So comparing even plain PDMS, which is it's a polymer, with a nanotextured PDMS, we could see there were many more captured cells for that. And even better than what you would get on the surface of a glass slide. So the glass slide is a workhorse, right? So glass slide is where every histology is done on glass slide. So, but if you create nanotexture on a substrate on PDMS, we could capture many more tumor cells using nanotextured PDMS. So that's something which leads to that original problem of finding those tumor cells in a given sample. But this one, this works then tells us that we can do this, and we have shown that this can be done with biopsy sample, where we can now look for and find the metastatic cells on top of just cancer cells in that chip. And hence, that gives us capability of early detection. So the cells that we would miss in standard microscopy detection, we would not miss if you are using a nanotextured substrate or nanotextured PDMS in this case. And not just that, if you do the incubation, we see those nanotextured surfaces, the cells really like it. They spread, they grow much faster and much cleaner on those cells. So these are 3 cells which were shown same time, same condition, but the substrate was different. And we look at nanotextured PDMS, the cells really are making a very good connection. So this dependence on nanotexture, on binding, we then prepare different nanotexture levels also, where you could change getting the roughness. You can get higher one if you do more etching. So we were playing with reactive ion etching here. So here the words average roughness is, we say AR, and R is just roughness. And what it is doing is, we are now finding with higher and higher roughness, we are capturing more and more cancer cells. And in this case, we are using a dye to look at those cells which are captured on the surface. Now, this is something which can be used to in a microfluidic channel to detect different colonies or different cells. So another work which we did was where we looked at brain tumor. So brain tumor, especially glioblastoma, which is very aggressive and very deadly brain tumor. So in this case, what we did was incubated the surface with the human glioblastoma samples and washed and saw that what are the differences in those surfaces going from one roughness to the highest roughness. And what we found was the number of cells were dramatically higher in case when we were increasing more and more. So the number, the red one here is, sorry, it's missing. The red data is for astrocytes, which are normal brain cells. So the data shows that the efficiency increased of binding. That now in this case, we can, from the background, which is the normal cells, we can increase it by 5 times, almost 4 times from that. And that's statistically different. We are improving the detection capability of a microfluidic channel, microfluidic devices that everybody is using. But what they're using is just a plain surface of microfluidic channels, the bottom, the walls. Here, what we are doing is we are incorporating another dimension, which is nanotexturing that. And that gives you many, many more cells binding and higher sensitivity and selectivity. And not just that, if you do the incubation and let the cells grow, they grow much faster on nanotextured substrate, as good as you would do on a cell plate. So cell culture plates are what we do in regular standard scientific studies. So nanotexture gives you as good of that, not just binding and selective capturing, but if you do culture, that's also much more for cancer cells with respect to the normal cells. So hence, this goes back to that original idea of using the nanotexture in metastatic cells to make a device that can not just bind them, selectively capture them, but give you a culture also, which definitely tells you these are tumor cells. And this was data about for 3 days, right? Now, living things are not just nanotexture, so there are different hierarchies of texture, micro and nanotexture. That's another thing where we said, okay, let's do a micro and nanotexture, kind of a mixture, just to emulate more of living systems where we can see the cell attachment that would happen in a real physiological system. So here they did some comparison between aptamer and antibodies also to verify the sensitivity of the surface. So in both cases, the number of cells captured on a hierarchical micro and nanotexture surface was much higher than compared to just nanotexture surface. So adds another idea of a nanobio interface, which gives you much more better binding with or selectivity against tumor cells. And again, looking at the data, we see that between the astrocytes and HGBM, we see much more binding for cancer cells than for normal cells. So this is the data for antibodies as well, which gives you a similar but slightly less efficiency than that. Now, again, going to the idea of looking at individual cells. So what did we do? We looked at those cells who were moving on the surface, and we identified some metrics of those images. And if you think about it, this is just an image processing that geotech people do. For example, they look at the lakes to find out how much water is there in the lake. So they look at the boundary from satellites, and that can tell you. So we thought about similar idea that a cell is nothing but a boundary, and the boundary is changing. So we can define a bunch of metrics and look at those boundary changes in terms of those functions that we can define to separate out normal cell from tumor cell. And that's what was done by, so this is just one example of one feature, which is Hausdorff distance. And there is a definition of that. So if you look at that over time for a cancer cell on an aptamer versus a cell on a surface which has a mutant aptamer where there's no binding happening, we see big difference. So these differences can be termed in terms of the radial distance, the average area that they are covering. It can be the boxing of the cell and looking at the area of that box. So a bunch of those. What we did that, we took them and we made an AI system. So we are taking the data already. So we are looking at the cell behavior, and then we said, okay, let's translate that behavior of a mutation of healthy cell. How does it translate into some metrics that can be used with machine learning? So the idea is taking the biological differences and translating into functions which can be measured in an automated system. And for that, we need to define some parameters and look at do the regression of the data and look at how it's changing. So what we saw that was, it's an overall idea of what we did that. So we got those features, we extracted those features, and we trained the system. And then we took the data of unknown samples and used that predictive model. So what we are doing is we are using the cells on a functionalized surface, right? Nanotextured surfaces. And we take time-lapse images to record the behavior over 15 minutes duration. And then we take the features of those cells in those images to quantify we call them gesture, right? So gesture, like I'm moving hands and making gestures. And those cellular gestures can be were used to train the system and to build a database. And that training database then resulted into quantifying this behavior of cell binding that we were talking at the earlier slides, right? So it's the same picture at the lower level. But now what we are doing is we are looking at these cells. They do not look different, right? They behave different. And that's where the cell motility or cell ruffling, which is naturally for tumor cells in the upper level. These upper strip is for tumor, lower strip is for healthy cells. And that shows us the behavior or difference in the gesture. So gesture idea is something which in sign language, there are computer systems who can read sign language, right? There were even a game system also a few years ago, Microsoft Kinetic, who were looking at people and how you move, and that was the kind of way they were looking at the gestures. So we applied the same idea here, where we do gesture recognition, and this is where then we take this data and we use those features from the cells to look at, to quantify their behavior and to distinguish between normal cells and cancer cells. So here are few features, and there we have identified 60 features in the system which were published in this paper a couple of years ago. So now here we have cells, one cell, two cell, hundreds of cells. And then we, for each cell, we have their behavior or gesture functions. And then for each cell, we look at those features for thousands of data points. So it's a really big data. It's a big data problem, but as they say. But what we are doing is we are controlling the binding at molecular level. We are looking at the behavior of cell based on their EGFR binding on a nanotextured surface and using that to identify one cell from the other. So if you now, these are 9 features that we are showing just physically, what do they mean? So you can see this in this picture. There is, if you compare green to green, you can see from the left one to the right one, there is a difference. If you compare the yellow one with the yellow one, you see there is a difference over time between those two. And that's where we, it's just live data between two, where we can in real time look at those cells that are captured and distinguish one from the other. So I was talking about microfluidic channels. So nanotexture in microfluidic channels is something which we have pursued, where we can use the texture and see how would that change the cell binding, or how it would change the difference between one type of cell versus other type of cell. So here the idea was there's an inlet on the left side where you are putting all the mixture, blue and red, good and bad cells. And we did this with cancer cells, this work, and you push them through, and then you have another outlet, and you have 2 electrodes. You are measuring each cell that's going through, and you are measuring each cell that is going out. The difference clearly tells us there are cells that got captured because they were cancer cells, and they got captured because they come in contact with the nanotextured functionalized device, and that shows you the difference, and in real time can tell you if there were tumor cells in a given sample, right? So, and this just, and we played with the flow velocity, because that changes the selectivity and sensitivity if you flow things too fast. So here we are showing if you flow fast, then we miss the difference between cancer cells and white blood cells. I'm sorry, the text is overlapped. But the y-axis is the efficiency. So that's another thing where we had to adjust the flow velocity to see the cell capture efficiency. The higher the flow rate gives you more shear stress on and that reduces the cell capture probability for both types of cells, for that matter. So in this case, what we did was we separated out brain tumor cells from white blood cells. So again, some work where you can, in that capture region, we can separate out the 2 types of cells based on their inlet and outlet data. There is much more data, but I think we are close to 50 minutes and I want to leave some time. This data is showing the inlet and outlet microchannel, where you see that there's this cluster of region, which was the indicator of tumor cells in the inlet, which you see many a very few of them on the outlet. So these are the cell difference which got captured, and that shows an efficiency of 83%, which means that if there are 10 cancer cells in a sample, we would detect 8 of those. And that leads us again to that big aim of capturing all the cells that we can capture from a given sample. So before I conclude, there is the ultimate goal is that we want to cut down time and cost. We want to get the results faster, want to get the results cheaper, so it can we can not just deploy it in remote areas, we can also have people who can do these things in their own comfort of their own homes. So now looking at the situation with COVID, we even more than a year after the big outbreak, we are not able to have devices where we can detect infection at home, right? So this underlines the reason why these kind of interdisciplinary work needs to be done. So here are a few collaborators, students. Many of my students are professors, at least I think see 2 of those who are professors from this picture. And we have started, I'm not a founder, I'm just a servant, right? So founder are big people in big cars. So we're doing this thing, STEMatters, where we want to engage people from all over the world. And the idea is that we want, the science now is not restricted to one part or few parts of the world. Science is something that we all share together. And the inspiration is that we want to engage people, students from all levels, from all backgrounds, from science, technology, engineering, environmentals, and mathematics, to come together to find solutions to the contemporary problems. And how we do that? One thing is webinars, like right now we are gathered from all over the world. So I hope, because some parts are way behind in time, maybe they're not here. But the idea is that we should be able to engage people from all over the world in looking at and researching problems that can have big outcomes. So there are many outstanding scientists like Dr. Kaka. You say CA. What do you call yourself? Keka. So like herself, who are inspired and who can inspire many more people from around the world. So we do outreach and engagement programs, and we can think about doing some things at your campus or even in Malaysia as well. So that's something we're always looking for ideas and collaborations. And with this, I think I'll stop here. And if you have any questions, you can type it in the chat, or as Dr. Keka, she allows you to speak up, we'll go with that. All right. Thank you for your attention.